Okay. Uh, okay. Good evening, everyone. My name is Shomo. I'm the PMRFTA for this particular course, Computational Systems Biology. And this course is taught by Professor Karthik Raman, IIT Madras. Yeah. So last week, so this is week 9. So in the previous weeks, we discussed about different type of uh, analysis, like flux balance analysis, room, and other type of analysis. And we last session we talked about synthetic lethals and how you can use FPA to determine control versus a synthetic lethal condition and so on. So today we'll be expanding on that. We'll be talking about RFPA, which is the regulatory version of flux balance analysis, which basically is incorporating the regulatory constraints of various gene expression mm, caveats in your flux balance analysis. And we'll also talk about various use cases for this. So yeah, let's get started. Okay, so firstly, RFPA. What is RFPA and like why do we want to study this? So as I was saying, the RFPA is basically an extended version of flux balance analysis where you incorporate regulatory constraints, regulatory information, so that it makes up a better model to predict the increase or decrease of biomass depending on your objective function. Now, in FPA, we did not have these different regulatory constraints, right? Okay, so for RFPA predicts regulatory and metabolic steady states for short time intervals, computes regulatory state consistent with previous metabolic states. So basically, we are trying to make a connection of the total metabolic flux in the system with the gene expression. So yeah, so we are trying to incorporate the regulatory constraints along with basic FP modules. So basically, we are incorporating gene expression and other kind of information which can be a proxy to regulatory information to see how the metabolic fluxes change if we incorporate that. Because in a system, there will be the genes that are involved, the metabolites that are involved in the whole process are actually being governed by gene expression of their particular respective genes. So the genetic constraints that we have, those are very valid in a real life system of metabolic conditions, right? Okay. And after incorporating the various regulatory states, we use FPA. It's a simple FPA there to find flux distributions consistent with the current regulatory state, and it provides qualitative insight into transcriptional events and an integration into with metabolism, and it also offers a glimpse into cellular behavior. Like, for example, there's a very good example of growth of E. coli or differential growth, I might say, uh, of E. coli in a medium containing glucose versus a medium containing lactose. So basically, it's the whole lac operon thing. So if you recall what is a lac operon, and like if you think about the whole situation, there's the metabolism involved, but how and when this metabolism goes about is based on the environmental cues. So either you have glucose or lactose in your environment. Depending on that, certain genes will get activated or deactivated, and your metabolism will go in a certain direction. So this is a classic case where you can apply RFPA, where basically you are incorporating these genetic gene expression constraints, and then you can calculate the flux balance analysis. But there are certain limitations. The limitation is that it provides qualitative insight into transcriptional events, but it does not provide quantitatively how much increase or how much decrease it can happen due to this regulatory constraints. Right? Uh, okay, so if you so here I've used the term SRFP. It's basically a steady state version of RFP, regulatory flux balance analysis, because when we do the FP thing, we all assume steady state behavior. If you remember, S dot V is equal to zero. It's the steady state behavior of a system. So that is what we assume. So here also, We assume that, so that is why the name steady state regulatory flux balance analysis, right? So, yeah, if you apply this technique to study the metabolism of E. coli, uh, there has been this particular study by Rupin and co-workers, and they what they did was they analyzed the effects of metabolic and regulatory constraints on the whole metabolic behavior. While metabolic constraints were found to determine the flux activity state of a majority of the genes, the role of transcriptional regulation was also found to play a role in determining state of about 30 to 20% of the genes. So this is a work that demonstrates that, yeah, regulatory information is very essential because it can explain a lot of metabolic behavior, right? And so this is a schematic. On the right is a schematic representation of an integrated regulatory and metabolic network. So on the lower panel you have the metabolic network. On the upper panel you have the gene regulatory network of various transcription factors and genes getting controlled. The expression getting controlled by different factors. Okay, so so here also the idea is to maximize your biomass depending on your objective function, but generally it is to maximize your biomass. So yeah, you have these particular metabolites, one, two, three, four, five, six, seven. These seven metabolites here. On the left is the growth medium. So for example, in E. coli, it can be lactose or glucose, depending on that your metabolism will change. But yeah, so on the left is your growth medium. On the right is your biomass production. Basically your growth medium is getting in and your biomass is getting out from here. And all this activity of the different metabolites, the various reactions that these metabolites are involved in the system is actually governed by the gene expression. Right? So you have transcription factors that are governing certain genes. Those genes are making these metabolites that are involved in these reactions. So the whole process is tightly regulated and tightly constrained, right? Yeah, so the regulatory network component consists of set of interactions between transcription factors and other transcription factors and genes, as shown here. And the metabolic network component consists of a set of biochemical reactions between metabolites available from growth medium as input, and a pseudo metabolite representing biomass production as output. So, yeah, and so how they are represented here is basically your input is your growth medium, which is the glucose or lactose, and your output is the biomass production. But technically, in a system, you won't see like the output as a biomass production. But yeah, this is taken as a proxy uh, to the total metabolism that is going on inside the cell, right? The system, I should say. So the regulatory component affects the metabolic component through expression of proteins that catalyze the biochemical reactions. So basically, these are proteins and enzymes that are formed by these genes, and these enzymes actually control the whole reaction process of the metabolite. So um, yeah, and so they are controlled by these regulatory components. And the metabolic component affects the regulatory component by the activation or inhibition of transcription factors via presence of specific metabolites. So, for example, this particular metabolite 4, is actually activating a particular transcription factor, which is activating a gene, which is actually producing a protein, which produces an enzyme complex, which actually helps in the production of this particular metabolite. So this is kind of a positive feedback loop. So the more the production of this particular metabolite, the more activating it is on the transcription factor, and the more the transcription factor is activated, the more gene expression occurs, which in turn forms the enzyme that actually is driving the production of the metabolite. So this acts as a positive feedback loop here. So this kind of situations can also happen in real life systems. Okay, so this is actually a schematic representation of the whole flux regulatory or regulated flux balance analysis approach. Uh, and you can actually generalize this concept to integrated FPA, also incorporating ODEs to simulate the regulation. So the regulation that we are checking here, these are like simple regulations, like either it is increasing or it is decreasing. But technically, it is not that simple, right? So there's a Hill equation involved. There is There are other parameters involved on how a particular increase of a concentration of a particular transcription factor can actually increase the gene expression. It's not a one-to-one. So twice the transcription factor concentration meaning twice gene expression. It's not like that. So there are constraints on that as well. So yeah. We can model those constraints as ODEs and incorporate them in the flux balance analysis to actually get a better model out of it. So let me go through the schematic. So first you start with a set of initial conditions and you derive the system state in different under different conditions, and then you include the regulation or the signaling part, which is the whole GRN or gene regulatory network part involved in the metabolism. So that you use Boolean expressions or you simulate an ODE model, and then you determine the constraints. Not determine, then you like kind of incorporate the constraints, which are uptake rates and on and off reactions, whatever, and then you do the FP optimization. So here the extra part here is the regulation of the signaling part. Also, you're incorporating in the form of differential ODEs, basically, or Boolean models, right? So yeah, and then you do the FP optimization for the whole metabolism part, and then system momentum flux, you get the system momentum flux values, and you update the system state for the interior and exterior conditions, and go on in a loop. And in between also you can incorporate the time series data as well, concentrations and growth rate, which can make the model even better. So yeah, so this is a rough schematic of how RFPA works. So basically, the extra part is here, where you're incorporating the regulatory component in your model. Okay, so now we'll talk about an example about how actually this SRFPA works. So, I'll take a specific example and we'll go through it basically. Yeah, so this SRFPA, is an optimization method that finds a consistent pair of metabolic and regulatory states. And then how do you solve uh, the basically your cost function or your objective function is through linear programming, mixed integer linear programming methods, and you form interdependency between metabolic and regulatory states using linear equations, primarily ODEs. So you have on the left is your regulatory state, uh, which you can have conditions. So gene 1 will be expressed when gene 2 is expressed and gene 3 is not expressed, or both of them expressed together, or something like this. So you have Boolean values. Actually, these are not actually continuous values because you're not modeling this as an ODE. So these are Boolean values. So either it can be off and on. So 0 and 1, right? So on the left hand side, you have the regulatory state. Uh, on the right hand side, you have the metabolic state where you have several of these reactions involved in the whole process. So these reactions are given by V1, V2, V3. And you can make the stoichiometric matrix out of this, which is S dot V. S is your stoichiometric matrix, V is your velocity, and at steady state, this is 0, right? So S dot V is 0, which is a steady state condition. And also on top of that, you have the regulatory information incorporated as well. So there are two kind of caveats to it. So one is uh, how is the regulation controlling the metabolism? And also the opposite question is true as well. How is the metabolism how is the metabolism controlling the regulation? So we'll see it from both ways. So yeah, so this is a regulatory and metabolic state. Now, the activity of each reaction depends on the presence of specific catalyzing enzymes, right? And for each reaction, you define a Boolean variable, which is Ri, specifying whether the reaction can be catalyzed by enzymes available from the expressed genes, and then you formulate the relation between Boolean variables Ri and flux through the reaction I. Yeah. So you can create the relation between the Ri, which is basically the reaction that is catalyzed by enzymes available from the expressed genes, and the flux through the reaction. So this is the whole module. So you have three genes involved here: G1, G2, G3. G1 is creating an enzyme, and G2, G3 are creating two proteins, which are forming a protein complex together, which is forming another enzyme. And either of this, basically, enzyme 1 and enzyme 2, can actually form, like control this reaction, or basically help the reaction to proceed. So the condition here is actually G1. So G1 is getting expressed and it is forming the enzyme 1, which is enough for it. And then you have to have G2 and G3 to form the enzyme complex 2. Now, enzyme 1 and enzyme complex 2, they can be present one or the other. So this is the condition that is given: G1 or G1 and G2. G1 or G2 and G3. Right. So this, I hope, is clear through. And yeah, so this is your regulatory condition. And now the presence of certain metabolites will activate or repress the activity of specific transcription factors. So we are talking about, sorry. So we first talked about this part, how the regulatory network is influencing the metabolic network. Now we are talking about how the metabolic network is actually influencing the regulatory network. For each such metabolite, you have to you define a Boolean variable specifying whether it is actively synthesized, which is used to formulate the transcription factor regulatory equations. Say, for example, one example let us take. So these metabolites M1, 2, 3, and 4. Now, this third metabolite M3 is actually helping the formation of TF2, right? So, which is a transcription factor which actually will which will help in expression of one of the reactants of this metabolic reaction, right? Excuse me. Yeah. So this particular metabolite M3 is actually helping the formation of TF2. Now, how would you write the equation for TF2? So TF2 will be expressed when TF1 is not expressed, because TF1 is actually inhibiting TF2. So not TF1 and MET3 or TF3. So understand the logic here. So basically, you have to in increase the production of TF2, or like you have to allow TF2 to express. When will TF2 be expressed? When TF1 is not there, so not TF1, and so this is not an OR, because both of them are acting together. So this is not an OR condition, this is an AND condition, right? So and either MET3 should be present to help express TF2, or TF3 should be present to help express TF2. So this actually becomes the equation for the transcription factors, or the genes involved, and this on the left becomes the equation for the enzymes involved. Right. So now if you do predictions on this, like if you create the whole model and predict gene expression variations, so the prediction of expression states changes between aerobic and anaerobic conditions are in agreement with experimental data. Right. So this is, I'm talking about a particular study that has been done on like basically different aerobic and anaerobic conditions, and how the different metabolic fluxes change. Now, the prediction of metabolic fluxes values in glucose medium are significantly correlated with measurements via NMR spectroscopy. So basically, your metabolic flux values in glucose are consistent with the experimental value that you have. Now, if you apply SRFPA on this, so SRFPA, if you apply on 103 aerobic and anaerobic growth media, so intermediate variability was undetermined expression or activity state in a given media. And in it, so this is the intermedia variability, and also there can be intramedia variability as well, which is variable expression or activity states across the say across media, different media. Now, a very small fraction of genes show intramedia variability in expression. A relatively high fraction of genes show intramedia variability in flux activity. So this is actually very important. So we have very small fraction of genes which show uh, intramedia variability in expression. So which is basically variable expression or activity across different media. So genes are generally gene expression is generally consistent across that, but a relatively high fraction of genes will show intramedia variability in flux activity. So the expression, in terms of expression, it won't change much if you change the media, but in terms of the flux, it changes a lot. So gene-like expression is likely to be more strongly coupled with environmental conditions than reaction flux activity. So this is what they actually found. So these are the results for that. So you have transcription factors, so express, non-express, non-determined, and intermediate variability. And also you have uh, transcription of regulated TF regulated genes, non-TF regulated genes in active, inactive, and non-determined conditions, showing intermediate variability. So this is the results from actually, I think, E. coli, from what I remember. But yeah, so these are actually different conditions where you can apply this SRFPA and get good models and good predictions out of it. Okay, now we will talk about something called efflux. One second. Yeah, now we'll talk about something known as efflux. So in the last part, we talked about SRFPA, which is basically incorporating regulatory information in your metabolic model, metabolic network. Now we will try to incorporate gene expression in the metabolic model, right? So it's basically a similar concept to get more information and make the model more robust. So yeah, this is illustration of an efflux method. So the core idea of the method is that we use gene expression to set maximum flux constraints on individual reaction. So the flux constraint that we'll put on individual reaction will be based on gene expression. Okay? How much of the gene can actually be expressed and what is the corresponding flux it can produce for that particular reactant and then and the enzyme or the reaction. So based on gene expression values, we put bars uh, on basically individual reactions, right? So yeah, so in this example, it is illustrated as pipes of different widths around each reaction. So basically, this means there's a high gene expression. This means there's low, so like this, right? And here it is shown a simple model comprised of four metabolites. So there are four metabolites A, B, C, and D, and four internal reactions. So these four metabolites involved in these four reactions, an uptake reaction for A, and a reaction converting the final product into biomass D. Right. On the left are simulated gene expression data for four genes whose enzyme catalyze four internal reactions. So green is basically lower expression, and red is corresponding to higher expression. And on the top panel, in the top panel, which is in the first gene. involved in this. So G1 is poorly expressed, and this can be conceptualized by this thin, uh, basically line. So G1 is contributing to this particular reaction, G2 here, G3 here, G4 here. So the width of the pipe is actually proportional to the expression value. So here the expression is actually pretty low for G1. And in the bottom panel, actually G1 and G2, the expression have been bumped up. It is expressing more. So this pipe, actually the width of the pipe has increased. So here are two conditions. Actually, if you look at the condition, these are four genes, four reactants, and four metabolic reactions. And these four reactions are and governed by four enzymes, which are actually produced by these particular four genes. Now, in the first case, you do not have very good expression for G1 and G2, and this is the predicted flux for that. So in that case, G1 and G2 are basically involved in R1 and R2 reactions. So the expression is lower. So the final biomass for R1 and R2 will be, final flux for R1 and R2 will be lower compared to R3 and R4, because G3 and G4 are expressing more here. Now, in the second example, in this case, G3 and G4 are actually having lower expression compared to G1 and G2. The more red, the better expression it is, the more expressed it is, right? Okay. Uh, uh, yeah. So you can see the difference in the predicted flux depending on the gene expression. So the idea here is to convey the fact that gene expression actually has a very big role to play in terms of how the metabolic flux would be maintained in a system. Yeah. So unlike conditions in which uptake of A is not limiting, so that is also a limiting factor here, because if you reduce the concentration of A, the whole metabolic flux changes, right? So if you assume that all mm, the uptake is unlimited, so there is no limitation on A, uh, then uh, then it would predict more flux through R1 and R2 in the bottom panel relative to the top panel R3 and R4. That is what I was explaining. Like you have more expression in terms of G1 and G2 here, so your R1 and R2 will have more flux compared to R3 and R4. And if you compare this condition with the previous condition, your gene expression of G1 and G2 were lower, so you had lesser flux. You had lesser predicted flux through R1 and R2 compared to R3 and R4. Okay, now we'll come to a concept known as elementary flux modes. So this is something, yes, a lot of questions on your assignments will also be based on this. And also, this is actually very intuitive to understand. So what are these elementary flux modes? So these are non-decomposable steady-state pathways in metabolic networks. They characterize phenotypes, quantify robustness, or identify engineering targets, right? And elementary flux mode analysis is currently restricted to medium scale models, and as the number of EFMs explode at the next network size, it's basically uh, 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 exponential curve. So basically, increase in slight number of nodes in the in your network can hugely increase the number of EFMs inside the network, right? So, so in while getting elementary flux modes, we classify the metabolites into internal and external metabolites. So the internal metabolites less steady state concentration, and the external metabolites can act as sources or sinks for flux. Then you calculate the stoichiometric matrix, which is an n cross n matrix representing the metabolite reaction relationship, positive coefficients, meaning metabolite is getting produced. Negative coefficients mean metabolite is getting consumed. So for non-trivial flux vectors, satisfying is equal to zero. So basically, under steady state condition, this is your steady state condition. So basically, your steady state, your SV S dot V equal to zero is a condition that is getting followed. So what are these elementary flux modes here? So these are minimal, unique sets of flux carrying reactions. Two words, minimal and unique. Like, these two are important, and they cannot be reduced into proper substrates. So basically, you cannot break down the reaction into smaller reactions, right? But these are basically one continuous reaction, and it is unique, and it is a minimal route, which is shortest route. And it is essential for steady state operation. Now, how do you find this elementary flux modes? So the, the idea is to get the minimal at the important flux carrying reactions, right? Unique also. So you iteratively remove. So in a network, you start by iteratively removing reactions while maintaining steady state. Computationally, it is very demanding due to this combinatorial explosion. So basically, when I have a large set of combinations of what to remove, what to keep, what to remove, what to keep, like that. So you can have computationally, it is very strenuous. Right? And for an example, is the E. coli code metabolism model. And if you try to find EFMs in that, you will find millions of them. So there are a couple of complexities and challenges for this. So the first one is computational complexity, because yeah, it is an exponential kind of a thing where basically you will have millions and millions of EFMs to analyze. So that becomes a lot computational work and computationally strenuous. And also the scale of the network increases EFMs exponentially. So if you slightly increase the network by adding one or two nodes, the number of EFMs that increase will be exponential. So it becomes very difficult to calculate. And yeah, okay. So for that, I would like to give an analogy for that, right? So, sorry. So this is actually a very good analogy. So I've taken it from a source. I don't remember the source. I would mention it here, but yeah, I've taken it from. So you'll find this example on the internet. So basically, it's an analogy between a subway map and these metabolic pathways, right? So imagine two people here named Anna and Bruno. They're living in a city called Vienna, and sorry, I'll produce these names. I pronounce these names very badly, so forgive me for that. So Anna lives in Otterkring and Bruno lives in Heiligenstadt. Okay, whatever these places are, these two people live in two different places. And Anna wants Bruno to bring ice cream from another place, but Bruno wants the quickest route to Anna's place. So what is Anna's strategy? So she analyzes all possible routes Bruno might take, and she aims to ensure that Bruno passes through that particular place where to get the ice cream. So you have all this set of paths from one node to another node. Now we have to see how many of these paths are passing through a single node. If you remember correctly, this is one of the centrality measures of graph theory, right? So how much a particular node is involved in the shortest path between two nodes, right? So that is the closeness centrality. Uh, this is a relative metric that is used here as well, right? So now that this can be easily understood by the concept of EFMs, right? So as we discussed, the EFMs are minimal uh, reactions that go from one metabolite to another, or one metabolite to a to biomass, right? In a similar type of a way, in a similar type of an analogy, you can imagine these EFMs as minimal subway routes through the network. So you're trying to find, you have a set of roots and you're trying to find the minimal roots for this particular network. And then there are essential paths where any disruption means Bruno can't reach Anna. So that is how EFMs are defined, right? Now, Anna figures out that if a certain subway lines are disrupted, Bruno has to pass through this particular place where the ice cream is. With EFMs, she knows all possible routes Bruno could take. Now, if you think of the analogy, how do you apply this concept to metabolic engineering concept? So basically, you replace the subway stations with metabolic reactions, and the person with a carbon source, the person who is bringing the ice cream with a carbon source. Now, Anna's goal becomes efficient production of a product from a substrate, and the mirrors and so this whole thing actually mirrors metabolic engineering problem. So the idea of a metabolic engineering problem is that you are trying to maximize or minimize a particular biomass or a particular metabolite production or degradation, right? So here it's actually the analogy stands very nicely. So if you simplify the analogy, it's basically subway maps, they have pairwise connections between stations, while metabolic networks involve multiple reactants and products. But despite the complexity, the principle remains the same. Find essential pathways to achieve a desired outcome. Right? But that is the idea for both in this example and also in the metabolic system. I'll just give me a minute. Okay. Yeah. Now we'll talk about. So we were talking about EFMs, elementary flux modes. Now we'll talk about another type of special type of flux conditions. It's basically extreme pathways. So as the name suggests, extreme pathways of a metabolic network are irreducible set of vectors that define the basis of the null space of a network stoichiometric matrix. Every allowable solution to the flux balance equation of a reaction um, network in steady state SV equal to 0 can be represented as a non-negative linear combination of extreme pathway vectors. Now try to understand here. So from a matrix or from a vectors point of view or a linear algebra point of view, so your extreme pathways are basically pathways that are on that define the basis of the null space. So basically, null space of a set of vectors is the space which is at perpendicular to that particular plane, right? So that is the plane that is defined. So you have a plane that is defined for the metabolic network, and then you have another plane which is directly perpendicular to the plane, which basically another set of metabolic reactions, but they have nothing to do with this particular plane, right? So that is what the null space means. The null space, in the null space, the basis of the null space is basically the vectors that actually form the, the actually span the whole space. Like if you can define a couple, three, so for example, um, if you think of three-dimensional space, X, Y, and Z, so the vectors that s- form the basis of the whole three-dimensional space are 1 comma 0, sorry, 1 0 0, 0 1 0, and 0 0 1. So one is the x cap, y cap, and z cap. These are the three vectors that actually make up the whole basis of the three-dimensional space. In this case, you are talking about the basis of the null space of a particular set of vectors, right? So the basis of the null space of the vectors, meaning the vectors that are in the null space of the original plane, but they form the basis. So those vectors actually make up the whole plane, like that, right? That is from a or linear algebra point of view, right? Now, every allowable solution to the flux balance equations of a reaction network is dot v equal to 0 can be represented as a non-negative linear combination of extreme pathway vectors. So these biochemical, what are these pathways? What do these pathways mean in a biological and thermodynamic system, right? These are biochemically and thermodynamically feasible pathways that transform a selection of a given substrates to a selection of allowable products. Extreme pathways have been extensively used for the analysis of metabolic networks. Typically, such analysis used extreme pathways to define possible phenotypic states of metabolic networks under different simulation conditions, to identify network redundancy, reveal eigenpathways. Again, we're not going into the detail of linear algebra, but these are all kind of analogous to all the terms in there as well. So yeah, reveal eigenpathways that effectively characterize all relevant physiological states of a metabolic network. So modified version of these extreme pathways analysis have also been applied to regulatory network and signaling pathways. Such applications are still in their infancy and are important research topics. However, as the size of the network increases, the redundancy of the network, that is the number of different pathways that transform a given substrate to a given product, becomes more apparent, and the number of extreme pathways actually increase rapidly. Excuse me. Sorry. Yeah. Excuse me for a second. Sorry, guys, I'm having hiccups. Please don't mind. Excuse me. Sorry. Okay. Uh, so, yeah, redundancy also exists in small system, but this can be easily handled and even provide insights to legitimate alternative pathways. As the number of extreme pathways increase at a drastic rate, performing insightful analysis using these extreme pathways will become increasingly difficult. And the fact that the set of extreme pathways of a biochemical reaction network defines the boundaries of its convex steady-state solution space makes them a valuable tool for metabolic network analysis, right? And further, they emphasize alternative pathways that exist in a network which may otherwise be overlooked, and that can enrich the understanding of its possible physiological states. However, the increasing details included in reconstructed metabolic networks leads to combinatorial explosion and the number of extreme pathways in computation time. So, as I was discussing, that you have the, the number of extreme pathways actually increase a lot uh, with the uh, with the increase of these metabolic networks, and obviously the computation time becomes drastically high. Sorry, I'm just having some issues with hiccups. Okay. Now, where are these applications of these elementary flux modes, extreme pathways, elementary flux modes? Where is this applied? Yeah, sorry. Now, EFMs have become a standard tool for analyzing medium-scale networks, typically models of central metabolism, in which the computation time is actually feasible. And I have listed down 5 such examples where EFMs are utilized, right? Due to their support minimality, EFMs correspond to minimal subnetworks that perform a certain function at a steady state. Uh, Due to their support minimality, EFMs correspond to minimal subnetworks that can perform a certain function at steady state and therefore have been used to identify minimal conversion routes or cycles in a metabolic networks. So basically, to find minimal subnetworks that can take you from one substrate to one product, EFMs are highly valuable in those situations. Right. So you can find these EFMs and you can directly find a pathway from a substrate to a product. So they can also be used to predict metabolic network properties such as gene reaction essentialities, structural couplings, block reactions, etc. Because in your EFM, if you have a gene missing, then you can see key like if the particular reaction will go on or not. So these conditions of deleting genes or reactions, this can be like if they are essential or not to the metabolic pathway, sorry, can be determined using EFMs. Using AFMs, all yield optimal rules for product or biomass synthesis can be identified. Moreover, EFMs have been used to investigate metabolic trade-offs, characterize optimal solutions of enzyme allocation problems in metabolic networks with kinetic information. These EFMs are frequently used to identify intervention strategies for targeted modification of metabolic networks, including techniques such as minimal metabolic functionality, flux design, or minimal cut sets. Sorry. Most of these methods make heavy use of EFM property, which facilitates a directed search for suitable interventions by blocking undesired metabolites while preserving desired phenotype. Several approaches have been presented in the literature to decompose flux distribution into metabolic pathways to identify the contribution of certain modules to a given metabolic phenotype. Right. So this, sorry. Okay. So this is one such example where you're trying to find a drug to target in the mycolic acid pathway for in tuberculosis. So this is actually a very complicated pathway divided into four basic modules, and the fourth module is divided into three modules itself. So the biosynthesis of mycolic acid can be considered as made up of four subpathways: a production of malonyl CoA. I think malonyl CoA is here. So this is the first production here. Then fatty acid synthase 1 pathway, FAS1 pathway, which is this. Then fatty acid synthase 2 pathway, FAS2 pathway, which is this. And FAS1 products into, so this product goes into this, and this product goes into this, and this product is also distributed here. So this is kind of a very complicated kind of a pathway, but yeah, the products B and C, products of B and C are then, so basically, let me go through the pathways again. So four sub pathways: production of malonyl CoA here, then the FAS1 pathway, FAS2 pathway, and the condensation of FAS2, FAS1 pathway into D1. This is D1, D2, and D3. FAS1 and FAS2 are dependent upon the production of malonyl CoA from the first subset. The products of B and C are then converted into different mycolic acids in D. Okay. So if you can find actually the EFM in this particular whole pathway, what you can do is you can design drugs to target different uh, metabolites or gene products in that EFM, so that the mycolic acid production can be halted, right? So that the infection does not carry on. Yeah. Okay. Given the cell wall contains different types of mycolates in varying proportion, the optimal production of mycolates can be captured into two different ways. Number one, only the most important mycolate is produced, and number two, the known ratios of different mycolates are fixed. These are the assumptions that we will be following under. And then the flux distributions obtained from FP using map model and objective function C1. So this is in an unperturbed state. This is in a state where this particular thing is deleted, and another element is deleted here, and here the inhibition of this same thing happening. It may be noted that actually, if you compare the wild type with these deleted ones, so actually you can control the production of mycolate by deleting either PCA or INHA, right? So yeah, it may be noted that the lines joining the various flux points have been drawn to aid the discerning flux peaks clearly. Otherwise, they do not have any meaning. So individual fluxes are here. It's not a continuous thing. But anyway, so that is the idea. So one more such example is the lycopene production pathway. Excuse me. So lycopene is another substrate. So lycopene is another product mm, that is actually we are trying to maximize or minimize here. So lycopene synthesis begins with the condensation of key glycolytic intermediates in which is this G3P and pyruvate, and it continues in a nearly linear pathway. The genes encoding for IDI and DXS. So I think here DXS is this, IDI is here. Are typical targets for lycopene overexpression along this pathway. So you're trying to maximize the production of lycopene. In the engineered strain used in the study, the IDI, ISPFT, and the DXS genes are overexpressed. Identifying gene targets are also shown as they're connected to the lycopene production. And this is actually the net reaction for the production. now, sorry. Now, a total of 965 metabolic fluxes were calculated, such as to balance the rates of synthesis and depletion of 546 metabolites. Maximum, uh, maximize cell growth yield subject to a MoMA alteration and utilize glucose as the sole carbon source. Initially, maximization of lycopene production was set as the objective function to extract characteristic phenotype behavior. Simulation revealed a direct inverse relationship between stoichiometric maximum lycopene yield and growth yield, and a direct relationship between stoichiometric maximum lycopene yield, sorry, and glucose uptake. Therefore, these two relationships suggest the need to reduce the growth yield and maintain a relatively high glucose uptake rate to support enhanced lycopene production. So on the right is actually a condition where you're trying to scan for single knockout target identification. And the phenotype for every possible gene knockout was simulated using FP and MoMA as an additional constraint. The genotype phenotype plot illustrates the effect of single gene deletions on lycopene yield. So if you, for example, uh, delete this particular gene, the production fraction will be around 0.1017 at that point. So the plot is given like that. Yeah. Shown is also in silico predicted growth yield of these knockouts. A single knockout scan predicted 8 genes whose deletion yielded enhanced product synthesis while satisfying a minimum growth condition. Gene GLI-A, so this one, appears twice since its function can be classified both as an amino acid biosynthesis and also vitamin cofactor metabolism. The enzymes encoded by these genes are AC, CYO, ENO, GDHA, GLYA, and the several others present here. But this is actually the maximum. This deletion of this actually is a maximum production of lycopene. So yeah. Now this was the condition for single gene knockout. What about multiple gene knockout, right? So the process of sorry, the process of maximum sequential phenotype increase is illustrated on the left. So this on the green bars are all wild type. This is after deletion of GDHA, and this is after deletion of GDHA and ACE, right? So uh, yeah, a triple knockout construct based on the double mutant GDHA and GPNB was excluded as it violated minimum growth rate requirement, but other conditions were there. So on the other hand, predicted triple constructs in GDHA-ACE background continue to show an increase in lycopene yield. The path of maximal phenotype increase is given by the solid lines. So you see this line here, this line here. So there's the maximum increase there. Okay. However, since GDHA, GPMB knockout has been excluded for triple knockouts consideration due to growth rate, the next highest optimal path is followed. This gene indicates that novel gene targets arise as the genotype is altered as a result of gene knockouts. This is essentially evident in the case of TALB. Although TALB increases the production level in GDHA-ACE knockout background, it is detrimental in the GDHA only knockout background. So lycopene production and single and multiple knockouts in certain genes actually can uh, change the uh, biomass production. So yeah, that is what we are discussing here. And actually, this is experimental results for the single and multiple gene knockouts. So these are your knockout constructs: single knockout with these genes, double knockout with these set of genes, triple knockout with this set of genes. And this is the percentage increase in lycopene content. So GDHA actually is giving the maximum. A double knockout of GDH and AC is also there up there, but a triple knockout of GDH, AC, and TALB are actually the maximum, which is 19%. No, sorry, the GDH, AC, and FDHF giving the maximum, which is 37%. Sorry, the mutant growth rates are shown here actually. So this is the yeah. So this is what it is highlighting. So total lycopene content increases with multiple knockouts obtained along the path of highest production, with total lycopene content increases with multiple knockouts obtained along the path of highest production, with the exception of GPMA. So I think this, yeah, is actually decreasing lycopene, right? Overall, this data qualitatively shows the trends of simulations as predicted. Numbers, yeah, these are your SDs, so you have to account for this as well. But yeah, the idea is multiple gene knockout can increase your uh, lycopene production. Okay, so that is all in terms of discussion. Now we'll move to the questions. So I'll keep the uh, slide on screen for a couple of minutes and then we can discuss. Okay, so the answer here is Boolean expressions. So if you remember this example, this is actually a very good example for that. So you have metabolic states in and the different reactions given by V1, V2, V3, and these are the different regulatory states, 0 or 1. You can incorporate them in terms of differential equations as well, but this is a more standard way to do it. Right? So hopefully that is clear. Okay, so E stands for expression. So the answer is true. It is the constants on the bouts of a reaction are determined by gene expression levels. So yeah, that is true. The question is which of the following can affect can help to identify a potential drug target for bacterial disease affecting humans. So the answers are a gene that significantly reduces biomass flux of the bacterium when knocked out, which is understandable, so which is option number one. And option number three, which is a bacterial protein involved in biomass production of the bacterium, having a structure different from any human protein. So let's explore the other options as well. A gene that significantly reduces biomass flux of the bacterium when knocked out. So, sorry, was the correct option. The incorrect option, the gene that does not change the biomass flux. So there's no point in targeting that gene because no expression will change, right? A bacterial protein involved in biomass production of the bacterium having a structure very similar to a human protein. What will happen is like it'll like ambiguously target both the proteins and the the human side will also get affected. So you do not want that. You do not want the drug, sorry, the drug target actually to knock out human proteins or target human proteins which are target proteins that are similar to humans because then it can target the human proteins as well, which will not be ideal in our case, right? So yeah, moving on. Sorry. Okay, the systems level filtering for potential targets should include network analysis because you would want to know if you target if you actually knock out or knock down your drug target, what other genes and other metabolites are going to get affected. Flux balance analysis, obviously, this is understandable how the metabolic flux will change after knocking out or deleting or targeting a particular gene. Essentiality determination. So, is actually the the disease or the phenotype that you are targeting, is the gene essential for that phenotype? So for that, you need to do kind of an EFM analysis to find if it is essential or not. And sequence and protein structure analysis in terms of like drug designing, that is important, but in terms of finding the drug target, it is important, but in a different sense, not in the sense that we are talking about. Yeah. So the question is regulatory FPGA. It integrates information from gene regulation in metabolic networks. It can also be used to understand catabolite regression. Surely, it integrates data from micro experiments. It can. It cannot determine flux distributions in a metabolic networks. That is wrong because FPA can do that, and RFP is FPA only like adding a regulatory component to it. So it can. So that is not true. Right. Moving on. The question is how many EFMs are observed in the mentioned metabolic network, right? I'll give you one more minute. Two conditions. One, and let's talk about the first condition and then we'll talk about another condition. Okay, so you have to find unique and uh, end-to-end pathways, right? So, for example, the first one, R1 from A, then B, then R3. So this is one AFM, R1, R2, R3. R1, R2, then R5, then Rx is another EFM. R1, R7, R8, R9, and Rx is another EFM. And R6, R8, R9, and Rx is another EFM. Right. Now, these are four EFMs here. Now imagine the R4 is a reversible reaction. How many EFMs then? The answer is going to be five, one more than this. So this is going to be another pathway, R1, R2, and R4. Because this will be reversible, right? R4 will be reversible. So R1, R2, R4 will be another pathway. So five for if R4 is a reversible one, and four if this is the condition that exists. Okay, so final question. Okay, the answer is the expression of level of a gene. So the question is, match the upper bound constraints used in efflux with the types of gene expression involved in the reaction. So the expression level of gene, the bound constraint will be the single gene for a single reaction. Right? Sum of expression of all genes should be multiple genes involved in a single reaction independently, which are known as isozymes. And the minimum of the corresponding gene expression level would be multiple genes jointly required for a reaction. Okay, so yeah, that's it. So I'll close this session and yeah. Meet you next week.